Hi everybody, welcome to your Module 3 lecture. And today we'll be continuing off on what we previously learned, expanding on it, figuring out how we look into study design and how that applies to drug literature and evaluation. So within this module, we're going to be figuring out how to establish causality criteria. Again, continuing from the previous lecture of causation and correlation are not always the same. And developing an understanding of how we apply those to different study designs, and then differentiating between study designs themselves, because this is key for understanding what data means, and then how do we select appropriate study designs or methodologies to answer particular research questions or in the context of other constraints. So we talked a lot about in the previous lecture, correlation does not mean causation. So how do we actually establish causation? So there's a series of steps that we usually go through. First, you have to identify the variables. So what are the independent and dependent variables? We have to apply Bradford-Hill criteria, which we'll talk about. Evaluate the strength based on the study methodology. How good was that study? Was it designed really well? And then establish causation. Determine if true causal relationships exist between variables, how that is, how confident can we be in that, and what else can we do to see if they're truly causative or just merely associative. So what's that first step in the causal relationship pathway? It's figuring out what the dependent and independent variables are, and the way we try to remember this in this class is through the acronym DRY MIX, okay? And so why do we do this? Because a causal relationship exists when a change in one variable directly results in the change of a second variable. So a change in one variable, being independent, causes the change in the dependent variable. So how do you figure out what's what? Dependent variables are the dry part. So dependent variable equals responder. Dependent variable, responder variable measured on the y-axis. Dry, D-R-Y. It's changed by the independent variable. It's usually the variable that is measured, aka the outcome. And some examples of this we see in the literature we're going to be looking at are blood pressure, side effects, depression scores, things like that. Then how do you figure out if it's an independent variable? That would be the mix part. So this is the manipulated variable. Sometimes we think of this as the intervention. Therefore, it's the independent variable measured on the x-axis, MIX. It drives the change of the dry variable, the dependent variable. Is usually the variable that is manipulated, meaning that something that has to do with intervention. Okay, and we think about this in terms of our pharmacy studies as the drug. Amlodipine affects blood pressure, aspirin affects platelets. Once we've established the dependent and independent variables, we can then apply Bradford-Hill's criteria, which includes 9 different elements. First being temporality, the cause must precede the outcome. Strength, there must be a strong relationship between the variables. A high relative risk, for example. There must be some sort of gradient to the relationship, meaning a dose response, meaning more exposure leads to more outcome. So when you change one, it changes the other in a consistent manner, which is the next variable. The relationship is consistent in different studies and populations. It has specificity, meaning that there's a single cause for a single effect. You can't have a cause that ends up having 18 different effects, but the one you're interested in. It has to be a little stronger than that, okay? And a lot of these overlap together. Plausibility has to have some sort of biological background or rationale that can be conjured up to support the causal relationship. It has to be coherent, meaning that the relationship is consistent with previous knowledge. It has to have analogy, meaning the relationship is synonymous with other similar relationships. Have we seen this type of relationship before in this biological context? And finally, it can be supported by experimental research. So randomly assigned treatment changes the clinical outcome. Change in cause equals change in effect. Okay, so this is the Bradford-Hill criteria. It really provides that systematic framework for epidemiological clinical research to establish causal relationships. It's not, an absolute requirement. It's not the end all be all, but it's certainly going to help you understand if associations you're observing are more causal or less causal. Just to reiterate, it's just not that simple where you can check all the Bradford-Hill boxes and you know it's causal, because we know there's many causal relationships out there that might not meet all or even half of the Bradford-Hill criteria. Okay, for example, the relationship between smoking and lung cancer is pretty indisputable that smoking increases the risk of lung cancer, but it doesn't satisfy all of the Bradford-Hill criterion, especially the experimentation or the specificity, okay? And this is due to limitations in knowledge. Sometimes we don't know. Sometimes we can't understand coherence, plausibility, or analogy based on our current scientific knowledge. Okay, so we need to view causality on a continuum, meaning some things are somewhat causal, probably, and some things we can say with fairly high degrees of confidence that they're causal. Okay, and so you can see that example here in the lung cancer checklist. It checks, you can see temporality, strength, biological gradient, consistency, and plausibility. However, it doesn't have specificities, and we really don't know about coherence, analogy, and we can't do experimentation by making people smoke or not. Okay, but still, given all this, we are fairly certain that smoking increases your risk of lung cancer. Now I know what you're thinking, do we need to know all the Bradford-Hill criteria? Well, yes, you should, but for this course we're going to really hone in on 3 of them, being experimentation. So altering the cause alters the effect. This is the strongest criterion, aka a randomized controlled trial. Temporality, the cause precedes the effect, and individual association versus group association. Okay, so we're going to be really emphasizing and thinking about these 3 Bradford-Hill criteria when we look at associations. Okay, and this is the foundation of this course, but you should be familiar with all the other ones as well. Now let's transition over to study design and introducing you to study design and why it's important. The reason that it's important is because it's critical to evaluating research outcomes and what they mean. Study designs can be categorized into different characteristics such as their intent or how they're timed or their level of assessment, things like that. They can also be categorized into how one selects the study design. So meaning, are you going to choose your study design based on research question or resources available or ethical considerations? All these things come into how studies are designed. Okay, and not all research questions can or should be addressed using the same study design. And finally, study designs can differ or give different levels of evidence hierarchy, which we'll see many times in this course. It's useful as we begin to learn about study designs, thinking about how a study actually occurs. Okay, and it usually occurs through several phases, from initial planning to final interpretation, that we can see here. So phase 1 being, how do you plan the study? You need to figure out your question you're asking, generate a hypothesis, and then start designing the study around that hypothesis. And then you figure out what population should we target or select or form to answer and to address that hypothesis. And then you determine how you're going to allocate your patients or your population to groups, right? Because you have to have groups in order to compare to have meaningful data. And then you go ahead with the actual logistical portions of data collection and analysis, so gathering in information and analyzing the results. And finally, interpretation and extrapolation with statistics like we learned about in the previous class, like incidence, prevalence, odds ratios, absolute risk, relative risk, all those great things help you draw conclusions and apply the findings. So those are the phases of research. Let's take a little more look into how we might classify study designs. So you might classify them based on research intent. So why are they being done? Are they descriptive? Are they just describing some sort of disease state? Are they actually trying to look at some sort of outcome in an analytical way? So case series are descriptive. Case control cohort studies are analytical. What about their time orientation? You could classify studies based on that. Are they looking at the past, retrospective, or are they looking at the future, prospective? And an example of this, case controls are retrospective, randomized controlled trials are prospective. you could classify study designs based on level assessment. Are we looking at individual patients or overall patient populations? So you could think case control, cohort, randomized control trials are at the individual patient level versus cross-sectional are at a population level. You could think about study design based on investigator orientation. Is the investigator just observing what's happening? He's just an outside observer, or is he actually intervening and making patients do something through an experimental model? So case control cohort would be observational, just watch and see what happens. Randomized control trial, you're treating patients with drugs, so you're intervening, therefore experimental. And finally, you could look at study design based on group assignments. Though we could look on grouping patients based on their exposure or based on outcome. So based on what they have been exposed to versus based on their outcome. We kind of saw this with the Slytherin example in the previous lecture. An example of this would be cohort, which is exposure-based. Case control is an outcome-based type study. We'll be going over this over and over again, but this is to get your feet wet with study design classification. Let's drill down into these classifications even further. Research intent, descriptive or analytical. Again, descriptive being describing the characteristics of a case, describing what it was about. What's the phenomena of the case or a population? Without testing specific hypotheses about relationships between variables. You're just describing what's going on. Usually involves one patient or maybe a group of patients. Again, this is why we think of case reports or case series. And it typically doesn't include that statistical analysis because you're describing. Okay? Descriptive studies answer questions about what, not why or how, versus analytical studies are designed to discover relationships or associations between variables. Therefore, you are doing some sort of analyses and hypothesis testing between variables. Okay, and this involves usually two groups or more for comparison, right? Because if we have one group, then what are we doing? We're just kind of describing. So two groups helps us give those comparisons and those analyses that have meaningful interpretations. They employ statistical methods we've talked about in the previous course. And the examples here we've already mentioned are case control studies, cohort studies, randomized control trials. So basically everything that's not a case report or case series. And they are answering or attempting to answer questions about the relationships between variables and how causal they are. So how does research intent actually influence the study process? Because it's basically one of the first steps you take after defining the research question. You define the research question, and then you can determine whether you need a descriptive or an analytical approach, and then you go from there. Okay, so the choice between descriptive and analytical approaches should be guided by the research question. Okay, if you're just describing, that's descriptive. If you need to figure out what the relationships are and test hypotheses, that is analytical. Moving on to the next classification of time orientation, the two big ones we think of here are prospective and retrospective. Prospective means the investigator sits there and does something to the population or patients that separates them into the groups and then follows them forward into time. Okay, data collection occurs after the study design is established. Groups are formed before the outcomes occur. So this is all randomized controlled trials and prospective cohort studies. Okay, but you need careful planning of data collection and to minimize biases, and we'll be talking about this. Retrospective studies, on the other hand, at the beginning of the study, the investigator sits there and goes, I have this giant data center, and I'm going to look back in time to look at separating patients into groups and then looking at the data to learn about outcomes that have already occurred. So it's going backward in time. So you're a time traveler at that point. And you're relying on previous collected data. Both exposures and outcomes have already occurred. And so examples of this are case control and retrospective cohort studies. And why are they done? Because sometimes it's not ethical to do prospective cohort studies. But one of the main things is that the data is already there. We live in the big data world, right? There's so much data about us. It's all there for the pickings. So it's relatively quick and resource easy to do these types of studies. So what is the actual study process for time orientation? And this one's pretty straightforward because it's prospective or retrospective, right? So the study's created, the researchers establish a hypothesis and design, and then they form their groups. Are they forming their groups and then following them ahead of time? Or are they forming their groups and looking at data that's already there? Again, prospective means they're following them forward and they don't know what the outcomes will be. Versus retrospective means they're looking at data that already exists, forming groups, and then looking at the outcomes between those groups that already exist. Moving forward to the classification of level of assessment being individual versus population. Okay, mostly what we will be talking about is individual patient assessment. So investigators collect data on individual patients, not just a large group. Detailed data for each patient. This allows for analysis of individual variations. So we see this with basically, again, everything we're going to talk about in this course. Case reports, case control studies, cohort studies, randomized control trials, all of them. Individual level assessments provides more detailed information, but requires more intensive data collection efforts, right? Versus population assessment, we don't see this one as much, but investigator collects data on large groups of people with no individual data and just looks at trends at the population level. So it's aggregated data. It doesn't have individual characteristics or variables. We can think of this as ecological studies or maybe epidemiological surveys, which we'll look at here and there. Population level assessment allows for broad patterns to be identified, but really doesn't tell you anything about an individual or their risk or their variations or things that actually contribute to the outcome you're interested in. Just group trends. Now, what's the study process for a level of assessment? As you can tell, these are mostly very similar as we go. You form the research question, and then you determine whether individual or population data is needed. And that's pretty much it. You go from there, and then you do your methods of data collection based on individual or population data approaches. Your analysis is individual versus population data, and your interpretation will be individual versus population. So the key step of level of assessment is pretty much always occurring when the research question is formed. Next up in our classification scheme is investigator orientation. So where is the investigator orienting themselves to the research? And this is somewhat similar to the time orientation we already talked about. So they can be oriented in a experimental or observational way. Experimental, again, the investigators intervening in the study. They're assigning participants to treatment groups based on randomization or whatever the method may be. And then they're watching what happens to these patients after they've intervened in their lives. So experimental. The research controls the exposure or intervention. So the examples here are all randomized controlled trials and some quasi-experimental designs. And why do we do these? Because they're the strongest level of evidence that we can obtain, but they can have limitations that we always will have to think about, and we'll talk about in detail in future modules. Or you can be oriented in an observational way, meaning investigators are there just to watch and see what happens. Hey, I'm not here to intervene. I just want to see what happens with these group of patients, okay? They're observing natural processes. They're not manipulating it at all. And the examples here are case control studies, cohort studies, cross-sectional studies. Observational studies certainly can provide valuable insights. They're certainly cheaper and easier to do when experimental approaches are not feasible or ethical as well. But they have limitations in establishing causality, as you could think, because you're not intervening, so you don't know exactly how those associations fulfill Bradford Hill criteria. I want you to think back to those. What's the study process for investigator orientation? Again, comes down to defining that research question. Once you define the research question, you can clarify whether the goal is to establish causal relationships or just observe the associations. Okay, but some other assessments you might need to do are group assignments. Choose whether the investigator is assigning the groups or he's just observing existing differences. That might help give the investigator orientation. An ethical assessment. Is it ethical to have patients smoke and not smoke and see if they develop cancer? Probably not. So that's going to be an observational study. And then data collection just means that you are going to implement the appropriate methods for data collection based on whether you're intervening or you're just observing. So that's what guides the investigative orientation in study design. Our final classification is group assignment. And this only applies to observational studies because in randomized controlled trials, we're actually intervening in creating the groups. So for observational studies, you can either group by exposure, meaning that you're grouping defined on the presence or absence of a given exposure, like did they smoke? Did they take a medication in the past? And then you follow those groups forward to observe if there's outcomes. And so this applies to all cohort studies. So like comparing aspirin users versus non-users in bleeds in the future. So it's particularly useful when studying rare exposures or multiple potential outcomes from a single exposure. Or you can group by outcome, meaning you group defined on the presence or absence of an outcome. Do they have high blood pressure or don't they? And then you look backward in time to identify potential exposures. And so this applies to all case control studies, comparing those with colon cancer versus those without, for example. And this approach is particularly useful when studying rare outcomes, okay? Because you can't go forward in time and wait forever for a rare outcome. Instead, you find those rare outcomes and then go back in time. Now, what's the process for group assignment and observational studies? First, you need to select your population, right? You need a population to look at. And then you're choosing whether you're grouping based on exposure or outcome. And then if it's exposure, you look forward to collect outcome data. If it's outcome, you look backward to collect exposure data. And then you do the analysis and account for group assignment in the analysis. And here is a recap of all the classifications we just talked about. And I won't go over it in detail, but I want you to know this table well. Compare it to everything we just talked about. Make sure it makes sense. Okay, see where certain types of study apply to different types of classifications. You can see them all up there: randomized controlled trial, cohort, case control, cross-sectional, case reports, and see how they each have different characteristics for these classifications. Okay, you should know this very well. Now that we've gone through in detail the classifications for study design, let's go through the study designs themselves. So we'll look at case reports in series, case control studies, cohort studies, and controlled trials, randomized controlled trials, generally, that we've been talking about this whole time. Let's see what they're all about. Why is it important to know about study designs? Because there is actually a hierarchy of evidence, meaning how confident we can say causal relationships we are trying to determine, or at least associations, how confident we are when we are making decisions based on certain study designs. And so we can see here at the top, the pinnacle of all evidence is systematic reviews and meta-analyses, and you should know this pyramid because this is a common pyramid. Next comes randomized controlled trials, okay, experimental studies. Next followed by cohort studies, case control studies, and then at the bottom, or the least strong evidence, okay, because the strongest evidence is at the top of the pyramid, are case reports and expert opinion. So know this pyramid, you'll see it a lot. You'll see it lots in lots of other contexts too outside of this course. So where does this hierarchy of evidence, this study design, really start for the most part? It's probably with James Linds's scurvy experiment. We kind of most likely have all heard about this at some point in high school, but it's likely the first clinical trial by Scottish surgeon James Lind, and they're trying to figure out why are all these sailors, these British sailors, getting scurvy. And really the reason, the context was that scurvy was devastating sailing crews. They didn't have refrigeration obviously back then, so they'd eat all these more fresh fruits and vegetables and whatnot early in voyages, and then they would get sick as time went on. So he hypothesized that it was likely due to some sort of acid, and so he tested his theory by giving different sailors different treatments. So cider, vitriolic elixir, vinegar, 2 oranges and a lemon, seawater, and purgative mixture. And so it really represented a critical milestone because he systematically tested different interventions in order to test his hypothesis. And even though his findings weren't implemented till later, this was really a critical milestone. Let's start making our way through these different study designs. And just to note, we will be going over systematic reviews and meta-analyses at the end of this course in a different module. So let's start with the randomized controlled trials. What do randomized controlled trials look like? Essentially, you're identifying and recruiting eligible participants in real time based on inclusion-exclusion criteria, and then you randomize them, or you randomly assign them to whatever intervention or control groups you've established. You administer that treatment or that intervention according to a pre-specified protocol, and then you measure the results and the outcomes and compare them between the groups. And so it's considered the gold standard because of this randomization process, which helps minimize biases and confounding and makes the groups as even as possible, basically. And randomized control trials, as we just learned, are defined as, or classified, I should say, as analytical, prospective, and experimental. And we can see that here. It's the gold standard for causality because of this experimental investigator actively manipulates the independent variable. Okay, do they get the treatment or not? The cause precedes the effect because they are giving the intervention before measuring the outcome. And then there's individual association, meaning the data is collected at the individual level, which allows for direct comparisons. So let's apply this to Lynn's scurvy experiment. The population was British soldiers with similar conditions. Intervention was 6 different treatments, including citrus fruits. Comparison was multiple treatment groups, so he had 5 or 6 different treatment types we saw there. And the outcome was who got scurvy. And so this is why it's considered one of the first, quote-unquote, randomized controlled trials. Despite being the gold standard, there are some limitations and constraints to randomized controlled trials. First are ethical. Some treatments we can't give to patients if we know them to be harmful, okay? Like you can't make people smoke or drink alcohol because we know that it's not good for you. And so you can't invite people, even if it's not forcing, because nowadays we always invite people to participate. You just can't do that. Similarly, if you have a highly effective treatment, you can't randomize people to getting that highly effective treatment or some other treatment because you just can't withhold good treatment from people. So that's why often you'll see active treatment controls where instead of a placebo, you have the current standard of therapy as the comparator. Feasibility challenges with randomized controlled trials mean that it's really hard to do them. They're super duper expensive. It's hard to recruit people. It's hard to follow them over time. I've done my own randomized controlled trial and it's a lot of work. So they're just not feasible for really large population studies or long time periods and things like that. And sometimes you just can't randomize things. Like you can't randomize for genetic factors very well or demographic characteristics or environmental exposures. You just can't do that. So randomized controlled trials don't lend themselves well to those types of exposures. Next down the hierarchy, we have cohort studies. And so this one proceeds at first like a randomized controlled trial. So you're identifying eligible participants based on inclusion-exclusion criteria. You include them in your study, and then you group them based on past exposure status. You follow them over time to see the outcome, and then you compare those that had exposed versus unexposed. Okay? So cohort studies are technically observational studies, where you're comparing outcomes in an exposed and unexposed group. And as we learned about, you can classify cohort studies as analytical. They can be prospective or retrospective. They are observational, and they are grouped by exposure. In terms of their causality or ability to determine causality, they do have some value in terms of their temporal sequence. Okay? If we identify the exposure before the outcome occurs, that helps check off that box, right? And so we can say that's a strength of cohort studies. It doesn't have experimentation, right? Because we're not manipulating the exposures. We're just seeing if someone was exposed to something or not. And they also check the box for individual association. So we're collecting data at the individual level, which allows for direct comparison. So they get 2 of the 3 causality criteria assessments, so they're pretty good. And so if you applied it to our scurvy example, you could think of a hypothetical example where researchers might identify sailors known to eat lemons regularly and those who don't, and then follow them over time to see the scurvy rates. That might be how you would do a cohort study back in the old days. Cohort studies are powerful because they have a few key strengths. Like, they're really useful when the exposure of interest is rare, okay, or something that is just unethical to be applied in a randomized controlled trial. You can also draw comparisons between groups, providing individual level associations between exposures and outcomes. You can establish that temporal sequence, okay, ensuring that the cause precedes the outcome. You could study multiple outcomes from a single exposure, which, as we talked about previously, may or may not be a strength, but here it's listed as a strength. And you can calculate things like incidence rates and relative risks. However, cohort studies do have some weaknesses. They're not randomized, so you have to make sure that the groups are comparable. They're vulnerable to selection bias and how participants are recruited and assigned to exposure groups. Sometimes and often they require larger sample size and longer follow-up periods for rare outcomes. And despite not being as expensive as a randomized controlled trials, the cost can become quite high, especially depending on what you're looking for and how long you're looking for. And then finally, you can have some loss to follow-up, which is also a weakness of randomized controlled trials, especially if it's related to the outcome. Turning over to case control studies, these ones start differently than the other two. You're selecting patients with the outcome of interest to begin with, and then you identify patients you can compare to the outcome patients that are comparable. Maybe you're matching them on age, sex, and race and ethnicity or something like that. And then you do the exposure assessment. You look backward in time to see if they are exposed to something like smoking or a certain drug you're interested in. And then you do the comparison for the frequencies of that outcome between, or the exposure and the outcomes between the groups. So case control studies are generally classified, as we saw earlier, as analytical, retrospective, observational, and grouped by outcome. What about the causality assessments and their ability to do that for case control studies? Really, they only check off one box: individual association, data collected at the individual level. So case control studies are much lower on the hierarchy for evidence because their ability to determine causality is really low. However, they're pretty useful for rare outcomes, okay? So investigating rare diseases or outcomes, they can specifically select those cases and then look back in time to give you some useful associations and data that you can work from, okay? And if we apply this to our scurvy example, we would want to identify sailors with scurvy and those without controls, hopefully matching them pretty well. And then we would look at their dietary habits to figure out how much citrus fruit they consumed. We generally see case control studies in epidemiological research because of their strength for looking at rare exposures or uncommon diseases or rare outcomes, okay? Especially useful for exposures that we can't do ourselves. So things like pollution or toxicants and things like that. That's why you see it in epidemiological research a lot. Their strengths also include comparing groups or being able to draw comparisons between groups at the individual levels, which checks that box we talked about in the previous slide. You could study multiple exposures for a single outcome. They require fewer resources for sure compared to cohort and randomized control studies. And they can be pretty quick compared to prospective designs because you don't have to wait and go forward in time. You select your patients and then you're going backward in time through surveys or chart reviews to figure out if they have that exposure you're looking for. Weaknesses here are it's retrospective, so it's relying on existing data, which can introduce confounding or other biasing factors, and along with that, it's not randomized. It's often difficult to establish temporality so that the exposure precedes the outcome, and you can't really directly calculate incidence rates or relative risks, but only odds ratios, as we learned about in the previous module. As we approach the bottom of the pyramid, we're getting to cross-sectional studies. And so for this one, basically you're going to identify a population or representative sample of interest. Then you measure the exposures and the outcomes at the same time. Then you look at basically correlations in that data set, and you interpret it which mostly means you're identifying hypotheses for further research. And so cross-sectional studies are generally classified as analytical or descriptive and have a unique time orientation and are observational in nature. In terms of their causality or their ability to establish causality, cross-sectional studies are at the same level but still weaker than what we just learned about because only individual associations are the box that it checks. So you can't get any temporality and you can't get any experimentation. They do have usefulness in determining the prevalence of something in a population. And so they're really relatively cheap and easy to do. And so you can figure out how much is something occurring in a population. So often they're used for that. And so applying to our scurvy example, you could do a cross-sectional study of British sailors to figure out how many were affected with scurvy and how many reported eating lemons. You could do something simple like that, but then you can't make any more further conclusions from the data other than just how much something occurs. So what are some strengths and weaknesses of cross-sectional studies? They're relatively efficient to do, they're cheaper to do, they can measure prevalence or disease burdens in populations. They can give you that snapshot. They are useful for generating hypotheses for future research and getting that funding to do the more expensive research. And they can have the ability to study multiple exposures and outcomes at the same time. Some key weaknesses here are obviously you can't do experimentation because it doesn't check that box. You're just observing. You can't determine temporal sequences. And there's selection bias issues, and you might not actually capture rare conditions if you don't select your population right. So despite that, there is some usefulness to cross-sectional studies, but they are limited. Finally, at the bottom of the pyramid, we have case reports and series. And so what's happening here, this is describing, right? We're just descriptive and almost always retrospective. We identify a unique case or cases, we're reporting that. We're documenting thoroughly what happened with that patient and reporting in the literature to generate future hypotheses that can be studied in other study designs. In terms of their ability to establish causality, they don't check any of the three boxes we're interested in, and I apologize for the typo on individual association there. But they just don't check any of the boxes. And so what are they really useful for? In hypothesis generation. So they kind of give you an idea, maybe something's going on here. Let's study it in a cross-sectional study, and then we'll study it in a case cohort study and things like that. In terms of the scurvy example, a case report might document a single British sailor who consumed large quantities of lemons and then remained scurvy-free. So then you can say, hey, maybe if we did a intervention study where we gave sailors lemons versus not lemons, maybe there's something there to study more. So it generates hypotheses. What are some strengths of case reports and series? They're useful for really rare or new phenomena. They can serve as a first indication for drug effects or unexpected drug effects or therapeutic benefits for future study with generation of future or new hypotheses. Weaknesses are you really can't draw any causation. You can't draw any comparison between groups because there's no individual association. And they're highly vulnerable to publication bias because most journals don't want to publish cases, or we'd just be flooded with cases. So with all that said, how do you figure out which study type it is? And so there's a little flow diagram which might be useful to you here. You can start by determining, is it an experimental or an observational study? If it's experimental, then you can basically figure out if it's a randomized or non-randomized controlled trial. If it's observational, then you got a few more steps, right? Is it descriptive or non-descriptive? If it's descriptive, then you don't have a comparator group, so therefore you're on case or case series. If it does have a comparator group, then it's non-descriptive and it's analytical, okay? And then you got to figure out which direction the study is going. Is it going forward or reverse, right? Is it going from exposure to outcome or outcome to exposure? If it's going from exposure to outcome, you've determined the exposure and you're waiting for the outcome, you're going prospective cohort study. If you're going from outcome to exposure, then you're going back in time, that's a case control study. Or if you're doing them at the same exact time, then we know that's a cross-sectional study. What are the key concepts from this module's lecture? Learning about causality assessment, so what goes into evaluating the strength of evidence for causal relationships, how we classify studies, where these studies fall in the evidence hierarchy pyramid, and how you actually design the selection for certain studies. And with that being said, I just want to reiterate what to take away from this module and what you should be expected to know, because this is just going to be the foundation for the remainder of the course. Causal relationships, classification methods, and study design differences. These are going to be key through all the activities we do and on the tests too. So make sure you know these things and know them well.